Hey everyone, this is video number 6 for the immune system. And in this video, I want to start talking about topic is adaptive immunity and focus on the lymphocytes, T and B lymphocytes. So they're very important cells for adaptive immunity, and there are a number of important steps in development of functional T and B cells. So let's take a look at that. The antigen-presenting cells are also important cells in adaptive immunity, but I'm going to save those for the next video. This video will be talking about B and T cells or lymphocytes. And so just to review from last time, those are made in the red marrow by hematopoietic stem cells. And they have to, when they're first made, they're not fully functional in an immune response. So they have to undergo a process of maturation. And maturation for the B cells in the bone marrow, for T cells in the thymus. Then what's next? Okay, so here are the events in a life of a lymphocyte. And these are common with both B and T cells. The details are going to be different. So I made it kind of general what's going on. And as we move forward, we'll get more specific. So as I mentioned, they're formed in the bone marrow and they mature either in the marrow or the thymus. After they're mature, they are immunocompetent and they are transported by the bloodstream to the secondary lymphatic organs. Spleen, lymph node, malt, tonsils. Okay. In the secondary lymphatic organs and tissues, they carry out immune surveillance. That's their job. If in the process of immune surveillance, a lymphocyte binds to a foreign antigen, that causes activation of the lymphocyte. That's the first step in mounting an immune response to this foreign antigen. Activation causes what's called clonal expansion. The lymphocyte that binds to the foreign antigen, and only that lymphocyte, will be selected to expand in numbers. And it forms an army of identical lymphocytes, a clone, and they're all capable of recognizing and mounting an attack on that same foreign antigen. The effector response is what is the attack? What is the lymphocyte going to do to battle that foreign antigen? So that differs in detail between T and B cells. And one thing that some lymphocytes will do after activation is they will form memory cells, which they don't exactly participate in the attack on the foreign antigen, but they're sort of socked away in the body for later in case we're exposed to the same antigen another time. All right, so this slide is just reviewing the first two steps, formation and maturation. And I'm going to go in detail about what happens during maturation for T and B cells, but this is just where it happens, red marrow and the thymus. Okay. When a T cell or a B cell leaves the site of maturation and occupies secondary lymphatic organs, they're said to be immunocompetent cells. All right, so let me go through the steps involved in maturation for both T and B cells. I made a video describing that. So we'll do the video first, and then we'll come back and just reinforce that with some slides in the PowerPoint. Okay, the lymphocytes are the key cells in the adaptive immune response. So lymphocytes have to go through a process to become what's called immunocompetent, meaning they're capable of carrying out a immune recognition of a pathogen and then an immune response to get rid of that pathogen by a number of different ways. Okay, so I wanted to go through that process, and so what I've drawn on the board is the process of lymphocyte maturation. And first of all, formation happens in the bone marrow And this is a generic lymphocyte that comes out of the bone marrow, or when it's first formed anyway, put it that way. And it could become either a B cell or a T cell. If it remains in the bone marrow, it's going to become a B cell. If it leaves the bone marrow and travels to the thymus, it's going to be a T cell, okay? So the maturation process is different in those two locations. One is going to cause changes to this new cell, new lymphocyte, to make it a B cell. The other changes to make it a T cell. Let's talk about the B cell changes first. So the changes are maturation. What's going to happen to a lymphocyte if it matures in the bone marrow? Two main things. One is each cell is going to make and display on its cell surface a unique B cell antigen receptor. Okay? They're equipping themselves with a protein receptor. It's called the BCR, B cell receptor. And this is going to be put on the cell surface, and it's going to be able to bump into maybe some foreign antigen, pathogen. And if the antigen receptor recognizes this, meaning it can bind to it, then that's going to lead to, the immune defense, the activation of this immune B cell. All right, but each B cell does this uniquely. The antigen receptor, the B cell receptor, has kind of a Y shape to it. We'll talk more about that later on. But it's unique, and it's unique at the very tips, and the very tips is what's going to be binding to an antigen. Okay, so I've indicated on this slide is the basic structure of the B cell receptor is the same in every cell, B cell number 1 and B cell number 2. However, the binding site, the very tips here, are different. So red indicates one type for number 1, cell number 1, black, a different type for cell number 2. For any given cell, they all make the same B cell receptor, okay? But every cell is going to make a unique receptor. That means of the millions of B cells that you make, they're all geared to a different antigen, potentially recognizing a different pathogen, okay? So that's step number 1. Each B cell makes a unique B cell receptor, puts it on its surface, and then it could recognize an antigen. Second step, very important step, is negative selection. Now, these antigen receptors could bind to any antigen. That means some of them are going to recognize foreign antigens, but some of them are going to recognize normal self-antigens. Do we want B cells that can recognize normal self-antigens to go out in the body and start mounting an immune reaction against our normal antigens? No, we do not. So this step, negative selection, is going to remove those B cells that make a B cell receptor that would recognize our normal self-antigens. They're going to be killed by apoptosis. They're going to be subtracted. They're going to die. And what we have left then are an army of different B cells. They are each equipped with a unique antigen receptor. And these B cells are going to be released into the bloodstream, and they're going to take up residence in the secondary lymphatic organs, lymph nodes, spleen, tonsils, malt. They're going to take up residence there. The B cells that get released then have these properties. They're immunocompetent, meaning they have an antigen receptor in their surface. They started out not being immunocompetent, no antigen receptor, but now they're immunocompetent. They can, each B cell can recognize and bind to some foreign antigen. The other main property that they have is that they're self-tolerant, meaning they don't recognize normal self-antigens, because if they did, they would have been subtracted in this step, negative selection, okay? So maturation for B cells results in a host of mature B cells that are both immunocompetent and self-tolerant. All right, and then they're going to be doing immune surveillance in lymphatic tissues. Let's look at the situation for T cells. T cells also go through maturation, and the steps are a couple of additional steps. So T cells also make a unique T cell receptor. It's not the same receptor as B cells, but it is an antigen receptor. Okay, and they put it on their surface, so I indicate it by these double black lines here. For T cell number 1 here and T cell number 2, they're unique antigen receptors. The antigen receptor that T cell number 1 here has red tips, so it's different from the one that number 2 has blue tips. They're unique, okay? One thing that's different, T cells and B cells, is that T cells make a co-receptor molecule. All right, so T cells are equipped both with the receptor, that's indicated TCR here, the double black lines, and a co-receptor. There are 2 varieties of co-receptor. CD4, that's what I've indicated on this cell, or CD8, okay? The T cell receptor is always going to be with its co-receptor, okay? I'll explain the function of these later on. Third step, T cells are going to go through negative selection just like B cells did. If one of these cells has a T cell receptor that would recognize normal self-antigens, then that T cell would get subtracted. It would undergo apoptosis in negative selection. Then the final step for T cells is there's a positive selection. Positive selection is testing these T cells to make sure that they can bind to MHC. MHC, the special kind of self-antigen I mentioned in the last video. I'm not going to explain that now, but it's important for T cell function. So the T cells that get through this process, and by the way, it's not very many, something like 2% of all T cells that start this process make it to the end. The rest are subtracted either by negative selection or by positive selection. Okay? The T cells that get through are immunocompetent. They have an antigen receptor. They're self-tolerant. That receptor is not going to recognize self-antigens. And another feature of immunocompetence is the co-receptor is able to bind to MHC. Okay? T cells that pass this test, like B cells, are going to be released into the bloodstream, take up residence in the secondary lymphatic organs. And then your lymphatic organs then are equipped with functional, immunocompetent B and T cells. Okay. All right, just to go through that, there's a lot of detail on that in that video. So just to reinforce that with some slides, part of maturation for both T and B cells is formation of a unique antigen receptor. T cells, it's called the TCR, T cell receptor. B cells, it's called the BCR, B cell receptor. For any given cell, all of the receptors are going to be the same. Okay, one cell, and each cell will have 100,000 of these antigen receptors on its cell surface. But for any one cell, it's going to be the same, meaning it can bind to only one antigen, foreign antigen. Okay, but if you compare 2 different, let's say B cells, or if you compare 100 different B cells, they're all making a unique antigen receptor. They're all going to recognize different foreign antigens. Same thing for T cell receptors. Maturation of B cells requires them to make a unique antigen receptor and to display it on their cell surface. Because they're on the cell surface, they can bump into and bind any kind of foreign antigen in their environment. And that's what we want for immune surveillance to happen. Negative selection involves killing, removing B cells that make a B cell receptor that could bind to normal body self-antigens. We don't want them released, so they are killed in the bone marrow and never released. The remaining B cells now that pass that selection, they have functional B cell receptors that can recognize a potential foreign antigen, and so they're immunocompetent in that way, and they're self-tolerant. We know that they're not going to recognize normal self-antigens. Those get released, and those are the B cells that are going to carry out immune surveillance in the lymphatic organs and tissues. For T cells, some of the same steps are involved. Each T cell makes a unique antigen receptor. It's not the same molecule as in B cells, but it's similar. It's able to bind to an antigen. So they display these antigen receptors on the cell surface. Any given T cell has hundreds of thousands of these. And for one T cell, the antigen receptor is the same. Comparing different T cells, however, they're all unique and different. Okay? In addition to the receptor, T cells require a co-receptor. And they're either one of two types, CD4 and CD8. And the importance of the co-receptor is this is required for activation of T cells to mount an immune response. T cells, like B cells, undergo negative selection. In negative selection, T cells with an antigen receptor against normal self-antigens get killed and are never released from the thymus. Finally, T cells undergo positive selection, and this positive selection is to ensure that the T cell is able to bind to MHC antigens. There are cells in the thymus that display these MHC antigens to maturing T cells, and if the T cells pass that test, bind to MHC, then they are released from the thymus. If they don't, they are killed. Okay, so this is a pretty tough, I guess the vast minority of T cells do not survive this process. All right, so when mature, immunocompetent, self-tolerant T cells are released, like B cells, they're going to take up residence in the secondary lymphatic organs and tissues and perform immune surveillance. And this slide is just illustrating the steps in T cell maturation, positive selection, making sure that that the T cell is able to bind to MHC on the surface of a cell in the thymus. And if it passes that test, it survives. If it does not, it's killed. Negative selection is making sure that T cells do not recognize normal self-antigens. There are cells in the thymus that present normal self-antigens to T cells. If they recognize them, they get killed. If they don't, they survive. Final step, I didn't really describe it this way, but the final step is T cells end up with only one co-receptor, CD4 or CD8. The book describes it this way. They undergo selective loss. They start out with both, but they undergo selective loss of one or the other. So that's considered part of the maturation process as well. Okay, a little bit more about the two types of T lymphocytes that are coming out of maturation. So the ones that have the co-receptor CD4 are called the helper T cells. And the ones that have CD8 are called the cytotoxic T cells. So these two types of T cells have different functions in the cell-mediated immune response. The helper T cells are generally assisting the immune response, that of the B cells, that of the cytotoxic T cells, and that of the cells of the innate immune response. So the helper T cells are pumping up your immune response in general, and that's why they're called helpers. They are making your immune response even more vigorous. Okay, the cytotoxic T cells, having the CD8 co-receptor, are special cells that are specialized for killing abnormal cells in the body. And this could be virus-infected cells or cancerous cells. And the way they do that is they interact with an abnormal cell by binding to a specific antigen that the T cell receptor, antigen receptor recognizes, and then they release this chemical, cytotoxic chemicals onto the abnormal cell, which kills it. And this slide is just showing the end result of maturation for both B cells and T cells. B cells have this kind of Y-shaped antigen receptor, the B cell receptor, and the tips of the Ys are the antigen binding sites. So for any given B cell, those are all the same. For different B cells, these are going to be unique. Every B cell has a unique antigen binding capability. For T cells, the helper T cells have T cell receptor and a co-receptor. The co-receptor for a helper T cell is CD4. For the cytotoxic T cells, they have also a T cell receptor and a co-receptor. The co-receptor is CD8. Each T cell contains a unique antigen receptor. For any given cell, it's all the same receptor and the same co-receptor. Okay, well that's the main process of maturation. And in the next video, we'll talk about how a lymphocyte is activated when it recognizes and binds to a foreign antigen through its antigen receptor. See ya.